Today, for Lecture 17, I'm going to talk about the genetics behind the Genographic project. The Genographic project is a project run by National Geographic to map the ancestry and migration patterns of humans by collecting and sequencing DNA samples from hundreds of thousands of people from around the world. As you may imagine, ancestry is determined by SNP information and how similar SNPs are between different groups of people. For males, the focus is on Y chromosomal SNPs. For females, the focus is on mitochondrial DNA SNPs. I recommend watching the following YouTube clip that gives background on the Genographic project. First, some basics on human genealogy. The following is based on our current best understanding of human ancestry from a large quantity of both archaeological and DNA evidence, but the story is not yet complete, although the recent explosion of DNA data is filling in the gaps. It is believed that current humans, meaning us, arose from a small founder population of approximately 1,000 people in Africa. The descendants of this group later spread all over the world. This founder group existed about 100,000 to 200,000 years ago, or about 4,000 to 8,000 generations. Human ancestry analysis is usually performed via patrilineal analysis for males or matrilineal analysis for females. The idea is that we can follow changes in the Y chromosome for males, since the Y chromosome is passed from father to son, and mitochondrial DNA for females, since mitochondrial DNA is passed from mother to daughter. Remember, the sperm only provides DNA, and so that the mitochondria in the fertilized zygotes come completely from the mother, which is passed on to both daughters and sons, but only the daughters can pass this mitochondrial DNA on to succeeding generations. Human ancestry can be thought of as one ginormous family tree, or really two trees, one for males and one for females. At the root or top of the patrilineal tree is the founding male, referred to as Y chromosomal Adam, and at the top of the matrilineal tree is the founding female, referred to as mitochondrial Eve. All of us currently alive represents the nodes or leaves at the bottom of the tree descended from the root. The general intuition is that people who share SNPs are related. Remember our previous exercise that siblings share 50% of their DNA, meaning 50% of their SNPs, cousins 12.5% of SNPs, and so on. This can be extended to more distant relationships. The basic idea is to reconstruct this human family tree using SNP information to cluster people into groups called haplogroups. This is done through a technique called phylogenetic analysis. For today and Tuesday, I'm going to focus on patrilineal analysis since we are leading up to my ancestry. The exact same principles apply to matrilineal analysis. The Y chromosome has many advantages that lend itself to lineage analysis. First, it is shorter than the average human chromosome. Second, there is only one chromosome in every male, and that is passed on to all of the sons. With the autosomes, there are two homologous chromosomes, and either one can be passed on, which can confuse the lineage analysis. Third, there's no recombination to scramble the sequence. The only way the sequence can change is by mutation. Fourth, there is a fast mutational rate so that new SNPs are created. Finally, there are few functional genes on the Y chromosome, so there is less selection for certain SNPs. Let's consider an example. Here we have the complete family tree for all males. We start at the top of the tree with Y chromosomal Adam, and all males currently alive, such as me here at the bottom, and Stephen. The key point is that this Y chromosome from Adam is passed on to his sons, which is passed on to his grandsons, and so on for 4,000 generations, leading to us who are currently alive. Let's imagine that a new SNP at position 1000 is created in one of Adam's grandsons. So as this Y chromosome is passed on through generations, it is mutating at a certain rate. And so here we're saying that at position 1000, the Y chromosome, there's a mutation, and this mutation first appears in one of Adam's grandsons. So one grandson has an A at that position, and the other grandsons all have T. If I descended from this particular grandson, this A grandson, then I will also have an A nucleotide at position 1000. Whereas if Stephen descended from one of these three grandsons, then he will have a T at that position. As a result, these two groups are distinguished by this particular SNP. And so all of these people who have the A nucleotide at the SNP at position 1000 form a haplogroup. And we all descended from this particular Adam grandson. A haplogroup is a group of people that share a common ancestor and share a characteristic SNP at a particular locus, which is what we described here. Primary haplogroups are labeled by letters in the alphabet, for example A, B, C, and so forth. Creation of a SNP by a mutational event defines a branch point in the tree and a new haplogroup. So here we have an example of a haplogroup, and there's a new SNP being created, and that creates the ancestral haplogroup that continues on, as well as this other haplogroup right over here. And then a second mutation, creating a new SNP, creates two new haplogroups, A and B. Typically, when we represent the human ancestry family tree, we don't try to represent every individual, like me and Stephen and Ramiro and so forth. Instead, each node in the tree represents a distinct haplogroup. Each branch point represents the creation of some SNP in which one group has one SNP allele and the other group has another allele. Here we have the primary Y chromosome haplogroups, starting with Y chromosomal Adam, and we'll go over this tree on Tuesday when we go over my ancestry, and we'll find out where, to which haplogroup I belong among these different haplogroups. To construct this human family tree, one uses phylogenetic tree analysis. The basic idea is you start at the bottom of the tree. So all of these different groups are at the bottom of the tree. So these represent groups that are currently alive. Then you link the most closely related haplogroups that differ, for example, at one characteristic SNP to form the most recent common ancestor haplogroup one level up in the tree. For example, here we have haplogroup I and J, and they, for example, may differ at one particular SNP, and so we create this new haplogroup IJ, which is the common ancestor to these two groups. And you keep repeating this process, so this particular common ancestor haplogroup is similar to this one, K, and you merge them together to form the most recent common ancestor haplogroup, which is now IJK, which is here. And you keep repeating this process of merging haplogroups, and you advance up the tree until you reach Adam. One can estimate the mutation rate in the Y chromosome or in the mitochondrial DNA from calibrating when certain SNPs arose with archaeological data such as a mass migration. This mutational rate forms a type of molecular clock with which you can date the formation of the branches in the ancestry tree. And then as you advance up the ancestry tree to Adam or Eve, you can estimate when they were alive. For example, one can estimate that Y chromosomal Adam lived around 120,000 to 156,000 years ago. And when you do the same analysis on mitochondrial DNA sequences, that suggests that Eve lived between 99,000 and 148,000 years ago. In summary, it is likely that modern humans started as a small founder population of about 1,000 people in Africa, roughly 100,000 years ago. Conceptually, one can think that among this group was Y chromosomal Adam, from whom all current males have descended, as well as mitochondrial Eve, from whom all current females have descended. This represents the patrilineal and matrilineal family trees. The basic unit of these trees is the haplogroup, which is a group that shares a key SNP that defines a common ancestor in which the SNP arose. There is no assignment due this Tuesday. Instead, as I mentioned last Tuesday, I want you to spend at least 3 hours working on your term paper. Look over the updated instructions for the term paper, make sure you're using the genetic terms on the list of genetic terms, and work on your presentation.